everyone. Welcome to day 3 of our DNA lessons. I figured that instead of me talking in front of the class today, that I would give this to you and let you guys kind of go at your own pace and draw on your own. So do a little bit of individual work instead. I apologize ahead of time if you hear my children in the background because they're here. Okay. So today we're going to be talking about DNA replication and mutation. So we are now extending from knowing the structure of DNA and what it does to how it duplicates itself and how mutations are going to occur in that process. So the very first word that we're going to be talking about is replication. And if you look on your note sheet, that's the very first blank that's going to be on there. So when we talk about replication, our definition is the process of copying a DNA molecule to make another DNA molecule. Okay, so if you think back to mitosis and we think back to interphase where the chromosomes are duplicating, right? They're copying. Okay, this is what we're talking about. We're talking about replication. So they're going to be copying this DNA molecule and they want to make another one that is identical to the current one. Okay, they don't want anything to be different or to be wrong with it. They want it to be the exact same. So replication happens in 3 steps. It's not a very complicated process. At least we don't make it very complicated. So the first thing that happens is the DNA strand separates. There's a lot of like proteins and craziness involved, but the biggest thing I want you to know is that the DNA strand, instead of being a double helicase, okay, this protein goes right up the middle and it basically like unzips the nitrogen bases from each other. So it takes them, the puzzle pieces, and it pulls them apart. So now instead of having a nice double helicase, you have 2 separate strands of DNA. The second thing that happens is now new nitrogen bases are added to the 2 strands. Okay, so you have the 2, and then new nitrogen bases, the ones that are supposed to pair up with them, get added to those strands. So now instead of having 2 strands, you have 4 of them, making 2 identical strands of DNA. Remember that that's super important that they're identical because we don't want DNA to be different from one another. Okay, so the DNA strand unzips, pulls itself apart, and new bases are added. And so now there are 2 identical strands. Each strand has one of the original strands there. There's a video on replication a little bit farther on in the PowerPoint that does a really good job and a good animation of showing how this happens. Your first task today is going to be create a drawing that represents the 3 steps of DNA replication. So now that we've talked about it, now that you've watched the video, make a drawing that represents the 3 different steps of DNA replication. And when you are done, when it's in your notes, raise your hand and I will come sign off on it. Okay, so with DNA replication, we know that this is happening in interphase, right? Before every time that a cell is going to divide and make new cells. So with 3 billion base pairs that are needing to be copied, there are a lot of chances for things to go wrong, right? 3 billion base pairs pretty much every time a cell is going to be dividing, okay? So how likely is it? How likely is it that a mistake is going to occur during replication? I would say pretty likely. it's pretty likely that mistakes are going to happen. But remember, there's lots of junk DNA. Your body's really good about fixing things. So even if errors do occur, a lot of times they're going to be able to be fixed before the cell actually is made or goes through mitosis or meiosis or anything like that. So when a mistake happens, it's known as a genetic mutation. Okay, so on your sheet, it's a blank, and we want to fill in mutation. And a mutation is a change in the nitrogen base sequence. Okay, so our A's, our T's, our C's, and our G's. Okay, so it's a change in that sequence of a gene that occurs during replication. So it would be when the bases are pulling apart, right, and getting new ones added, mistakes are going to happen in that process. Okay? Most of the time, mistakes are caught, they're fixed, but sometimes if it goes undetected or if it's a really big change, like an extra chromosome, the DNA is going to be changed, and there's nothing that they can do about it. There are three main types of mutations that we are going to talk about. There are other kinds that occur, but these three are the ones that are going to cause the most problems and the most diseases that we are aware of. So the first one we're going to talk about, number one on your sheet, is called deletion. And deletion is pretty much exactly what it sounds like. It is like if you hit the delete button, right, on your computer. Okay, it is actually taking out a base pair. So when a base pair is removed from the DNA sequence. And you can write the definitions if you want, but really I think it's just helpful to have the examples written down. Okay, so for example, if we have TA, GC, GCAT as our DNA sequence, right, those are our letters, when DNA replicates and a deletion mutation occurs, it's going to look like this. We'll still have almost all of them. Actually, sorry, I'm going to do this in pen so it doesn't look quite so chunky. Okay, so we would still have our T and our A G and a C, and then it would be A, T. Okay, so we're basically canceling out an entire section or base pair of DNA. Okay, so deletion is when you're removing out one of those. So if we're going to practice this type of mutation, I think it's easy to see an example with just writing a simple sentence. So we're going to start with the sentence, the dog bit the cat. So if there was a deletion mutation, a base pair would be taken out of that. So I have represented a base pair with a letter. So instead of having the dog bit the cat, now we have the dog bit he cat. Doesn't make a whole lot of sense, right? The second type of mutation that we're going to talk about is called insertion. Okay, so number 2 on your worksheet for mutations should be insertion, and that's just when a base pair is added into the DNA sequence. Okay, so instead of taking one out, now we're adding a brand new one in that's not supposed to be there. Okay, so if we're going back to our examples, we're going to have our T and our A, our G and C, G and C. So everything looks fine until all of a sudden maybe we'll add one more GC and then the A and the T. Okay, so there's one extra one added in to the sequence. So I still have the same sentence for this example. So we still start out with the dog bit the cat. Okay, and then what we're going to do is we're going to add another base in. So we're going to add another letter in that's not supposed to be there, and it will change the sentence. So this one says, the dog bit the chat. Again, kind of makes sense, but not really. Definitely different from the original sentence, right? They're not identical. It's changing the meaning, which really is what happens with mutations. It changes the meaning of that DNA code. Our last type of mutation that I want to talk about, so number 3 on your list, is substitution. Okay, and so if you've ever played sports, anything like that, you know that when there is a substitute, right, you're taking out one person and putting another person back in, right? So in basketball, if you're going to substitute somebody, one person comes off the court and another person comes back on. Okay, so you're not changing the amount of letters like the other ones, you're just changing one for another. Okay, so when a base is replaced, excuse me, with another incorrect base. Okay, so if I did this one, I could have my T and my A, G and C, and then maybe I'll have a TA and then an A and a T. So this base right over here gets completely changed. Okay, so that sequence is nowhere near what it used to be. Okay, but there's the same amount of base pairs. So on our last sentence, I have, again, the dog bit the cat. So if we're going to replace a letter, you could have done it a lot of different ways, but I changed the very last one. So it says the dog bit the car, which completely changes the image in your head, right, of what this dog is doing. So again, when you think about mutations, it changes the whole instruction manual when there is a mutation. It's time to pause for task number 2. Get a note card from the front of the room and you're going to create your own mutation sentence. You cannot use the dog bit the cat. You have to use something completely different. Be very simple, okay? And then underneath it, make sure to include all 3 types of mutations. So how would that sentence change with a deletion mutation, with a insertion mutation, and with a substitution mutation? So what happens when one of these mistakes is made? Okay, what happens when a base pair gets changed? What happens when something gets deleted? There are 3 effects that mutations can have on organisms. So this is located on your worksheet. There is that little box on the right-hand side on the bottom corner. So there are 3 things that can happen when a mistake is made. The first one is absolutely nothing. Sometimes these mistakes have no effect at all. So sometimes it could have happened like on the junk DNA where it's not coding for anything. Sometimes it's just not big enough to cause a change in the organism. It could be a small mutation where it actually doesn't even change what the coding is. So sometimes mutations, you won't even notice it. And they probably happen quite frequently. Like we talked about before, with 3 billion base pairs, it makes sense that you're going to have a lot of mutations happening, but most of the time, there is no effect. Okay, the second thing that can happen when a mistake is made is the traits of an organism change, and it actually makes the organism better. So there is a mutation that happens that is going to allow the organism to do better in its environment, so wherever it lives. Sometimes these changes in DNA result in them receiving a new characteristic. Okay? For example, polar bears never used to be all the way white. Okay? They used to be brown. They kind of deviated from brown bears a very, very long time ago. But when one of the polar bears had this white mutation, that actually bettered their chance of survival because then they were able to camouflage themselves more in the snowy environment. Okay, so once one animal, organism, plant, doesn't really matter, has this mutation, then if they pass it on and more start surviving with that, that mutation is going to all of a sudden become normal. And it's going to become just a normal trait instead of a mutation. The third and final thing that can happen when these mutations and when these mistakes in DNA are made is probably the one we think about the most, and that's going to be something negative. That's going to be a genetic disorder. And that's a problem caused by mistakes in an organism's DNA. So usually genetic disorders are not going to benefit an organism. Some examples of these would be sickle cell anemia. So what happens with that is the blood cells are supposed to be this nice round shape so that they're able to get through the blood vessels in a very efficient way. Sickle cell anemia changes the shape of the blood cells, and it can be really dangerous for people. And then I know we've talked about cystic fibrosis before, back a couple weeks ago. And remember, that's when someone who their body doesn't make the correct proteins, so their lungs don't allow the salt to come in and out. So it actually ends up with their lungs getting that really sticky mucus on the inside, makes it hard to breathe. They're coughing a lot. So that is caused by a small mutation in somebody's DNA, and then they're not able to function quite as well as other organisms. All right, your third and final task for today is you are going to visit this website, the National Human Genome. It does a whole bunch of research on the human genome and different genetic disorders. You are going to scroll down. You're going to choose one genetic disorder to learn about. There is a pretty significant list. In Schoology, there is an assignment button. You can just type right in there. You write the name of the disorder that you picked, the common traits associated with it, and the type of mutation it is caused by. If there is not something specific about the mutation, I would maybe pick a different one, but it's usually pretty good about telling you what type of mutation it is or where it's going to happen on the chromosome. Okay, so you submit that in Schoology, and then you are done for today.